Hello, and welcome to the second half of Genome 361. My name is Leo Pallanck, and I'll be taking over for Dr. Maitreya Denham in all of the lectures till the end of the quarter now. So I'm hopefully you had a chance to see my introductory video. I'll try not to overlap too much with what I said in that video, but one of the things I did say that I want to remind you about is that my lectures vary somewhat in length from one lecture to the next. Um, if I was having this class in person, I would have to actually have all of my lectures fit into the hour and 20 minutes or so that we have allotted to us for this class. But some of my lectures are going to be a little bit longer, some are going to be a little bit shorter. On average, they're going to average about what it would be if it was in person. But I think that this variation from one lecture to the next is actually a positive thing for you guys, because one of the advantages of teaching online is I can cover each of the topics to the extent that's necessary. So a lot of times when I'm giving a lecture in person, I have to cut out material to make sure it fits into the time that's allotted to me, or I have to add some extra stuff that's probably not really necessary to lectures just to get it all to fit into the hour and 20 minutes each time I meet with you. But by doing this online, I'm only covering the material that I feel like needs to be covered for each lecture. So, okay, I did want to remind you of that. I know I said that in my introductory video. The other thing I want to say that's different than it would be if it was an in-person class is that I ask questions throughout my lectures. And if it was in person, I would pause the lecture and give you guys 2, 3, 4 minutes, whatever length that is needed to answer the questions in class. But in my lecture videos, I'm just going to say, hey, you can pause the lecture for a little bit and try and answer the question and then restart the lecture. So I'm only going to pause for a few seconds, just enough time for you to stop the lecture, think about the question, answer it, and then restart the lecture again. Okay, so that's all I really had to say. I don't want to overlap too much with what I said in my introductory um, introductory video. So just a little bit about the topic here we're talking about. So in all of my slides throughout my lectures for the second half, I'm going to list the key points that I want you to get out of my lecture right at the very start. So it's actually not a bad idea when you get through the end of the lecture to come back to the first page just to make sure that the key points are things that you really feel like you understand. I also include a figure at the beginning of each lecture that illustrates some aspect of the lecture. Sometimes it's easier for me to explain the figure at the start than others because it may not have any meaning until we get to the end. But I'll just say a couple of words about each figure at the start, and then when you come back to the beginning, you'll probably understand why I put that in there a little bit better when you get to the end of the lecture. So in this particular case, this is a picture of Alfred Sturtevant, who was actually an undergraduate working in the lab of Thomas Hunt Morgan, a very famous fly Drosophila geneticist. And Alfred Sturtevant actually did something very important in Thomas Morgan's laboratory. There was something puzzling about some findings that they were making at the time where they were seeing results that didn't match what you expected from independent assortment. And it was actually Sturtevant that first figured out correctly what was going on. And from that, he was able to actually make a genetic map. We'll talk more about what those are, that told us something about the arrangement of chromosomes along the length of a chromosome. This is actually the first genetic map that was actually created. It was created by Alfred Sturtevant. And part of the reason I'm pointing this out is just to kind of give credit to Alfred Sturtevant for this major insight, but also to remind you that undergraduates can actually make pretty important contributions, and if science is something you want to continue on in the future, it may not be a bad idea to seek out a lab to kind of do some research in. Okay, that's all I really have to say about this first slide. This slide is just to remind me, if I haven't already told you, that my name is Leo Pallanck. My email address is here. It's also in the course syllabus, which is online. And if you need to reach me for some reason, you can certainly email me. If you have questions about the course material, I think it's much better to use the Piazza chat format, simply because other students can then see your question and they have the ability to see the answer, and I think it benefits everybody that's involved in the course. If you watch my introductory video, you also know that I'm encouraging students to try and answer the questions. It kind of helps me to get to know you a little bit better. I monitor the message board very closely, so if I feel like the questions are not answered sufficiently or if there's more context that's required, I typically add to the question a little bit. But again, if you need to get a hold of me for some reason, feel free to do so by email. But if it's a question about the course material, some clarification that you need, I think it's best to use the Piazza format for doing that. All right, let's get started here. This is actually hopefully a review of the material you guys covered in the first half. And I'm going to ask you a question that I think you already know the answer to. If you carry out a test cross of a dihybrid, what do we expect in terms of the ratio of the outcome? So these are traits that Mendel actually studied when he was doing his experiments. They might have been traits that you guys talked about earlier in the course. It's not clear that Mendel actually ever did this cross. If he did do this cross, he certainly didn't report the results of this because they would have been somewhat confusing to him. But I'm not asking you to tell me what the actual result is based on what you would see if you actually did this cross, because there's a little bit of a surprise here in that. What I'm asking you to do is tell me what you expect the outcome to be based on what you've learned up to this point. So again, the traits are plant height in this case and seed pod shape. And if you take a dihybrid that's heterozygous for the plant height trait and heterozygous for the seed pod shape trait, and you cross that to a homozygous recessive, and the homozygous recessive phenotypes are short plants and constricted seed pods, you're going to see all four different offspring categories coming out of this. Some of the offspring are going to be tall with inflated seed pods, some are going to be tall with constricted seed pods, some are going to be short with inflated seed pods, and some will be short with constricted. So the question I'm asking here is, what ratio do you expect to see from this cross based on what you've learned in the first half of the course? What's the expected ratio from this sort of cross? And I'm going to give you three choices to choose between. And choice number one is you'll see a 9:3:3:1 ratio. Choice number two is a 1:1:1:1 ratio. And choice number three is a 9:1:1:9 ratio. So as I mentioned at the very beginning, I'm only going to give you a few seconds to pause the video and think about this and come up with the answer. And then you can just restart the lecture and I'll show you the answer. All right, so based on what you learned in the first half of the course, hopefully all of you chose option number two. You'd expect to see a 1:1:1:1 ratio. And why is that? Why do we expect to see that ratio? And it's easier to illustrate why that's the case by looking at the gametes that are produced by these two plants that you're using in your cross. All right, so if we look at the gametes that are produced by these plants, so the tall plant with inflated seed pods, because it's a dihybrid, it can actually produce four different kinds of gametes, right? So there's four different gametes that it can produce, and you expect to see these four different gametes produced with equal probabilities. For any one, it would be a probability of 0.25. And for the homozygous recessive, the test cross parent, you'd expect to see only one type of gamete being made, so the probability of seeing this gamete is 1. So, and then if you look at the offspring phenotypes from this particular cross, you're going to see the offspring phenotypes occurring in a 1:1:1:1 ratio. So the probability of seeing any one particular offspring category is 0.25. So why is that? So to think about this a little bit more carefully, let's look at the chromosomes, and we'll put these particular genes on the chromosomes so we can actually look at how the chromosomes are assorting during the formation of these gametes. And again, the reason we actually expect this outcome is because we expect independent assortment. That's what you learned in the first half of the course. So why do we assume independent assortment? So now we're going to look at the chromosomes here. We're going to put the genes for the plant height trait and the seed pod shape trait on separate chromosomes. And let's consider what happens during meiosis. So based on the allele orientations that I'm showing you here, if you go through meiosis, you're going to produce the two types of gametes shown below. One of them has the both dominant alleles, and the other gamete has both recessive alleles. But here's the thing, spindle pole attachment is completely random. So the spindle can attach in this way, but it's equally likely that the spindle can attach in the way shown on the right. So this arrangement is equally likely. So all I've done here in these first two images is I've just reversed the orientation of the plant height trait. So the dominant allele is on the left in the meiosis that I'm showing you diagramming here on the left, and the dominant allele for the plant height trait is on the right in the meiosis I'm diagramming on the right side. Okay, so let's imagine what happens when this second arrangement goes through meiosis and you produce gametes. In this particular case, you're going to actually end up with the dominant alleles in one of the gametes and the other dominant allele in the other gamete. So now with the meiosis that's diagrammed on the left, you're going to see half of each type being formed. But in the meiosis on the right, you're also going to see half of each type occurring. And half the time the meiosis will go as diagrammed on the left, and half the time meiosis will go as diagrammed on the right. So if you do the math, basically, so again, half the time the meiosis on the left will occur, half the time the meiosis on the right will occur, because again, spindle pole attachment to the centromere is random. And then you'll see half of the gametes being formed at each of these two types on the left, and you'll see half of each type in the meiosis that occurs on the right. And then if you do the math and add this all up, it turns out you're going to see 1/4 of each of these. So that's why we assume independent assortment, because you're going to see spindle pole attachment as random, and you'll see 1/4 of each type of gamete being produced. So if you guys thought that the proper ratio would be 1 to 1 to 1 to 1, based on what you've learned up to now, that would actually be the correct answer. But as it turns out, for this set of traits, the actual correct answer is in fact 9119. So, and again, this is based on something that you have not yet talked about in the class. So that would be a puzzle based on what you guys have learned up to this point. So now we're going to spend a little time talking about why that ratio comes out that way. And in a later lecture, I'll come back to this point to tell you why it's precisely 9 to 119. For now, I'm just going to tell you why it's not 1111. Okay? So as it turns out, genes on separate chromosomes sort independently. P's have 7 chromosomes. Here they are, 1 through 7. And for a lot of the traits that Mendel worked with, the traits, the genes that correspond to those traits are actually on different chromosomes. So for example, I think one of the traits you guys looked at in the class is the gene for smooth versus wrinkled peas, which is on chromosome 5. Another trait that you guys probably looked at was the gene, the yellow gene, which specified the color of the peas. That's on chromosome 1. And as it turns out, genes on separate chromosomes assort independently from one another during meiosis. But as it turns out, the traits for the plant height, which is also the L gene, and the pod shape, which is the I gene, these are actually genes that are on the same chromosome. They both reside on chromosome 4. And importantly, something I'll come back to later in the lecture, they're actually very close together on chromosome 4. That ends up being important as well. So because they're on the same chromosome, they don't actually, this is not a given for all genes that are on the same chromosome, but in this case, because they're on the same chromosome, the genes don't necessarily assort independently. So let's talk about what happens when genes are on the same chromosome. So the way I pictured this initially was I showed that the genes were on separate chromosomes, but as I just mentioned, the plant height and pod shape traits are actually on the same chromosome. So I'm going to move this up to the same chromosome and go through this meiosis again to show you what happens when genes are on when a pair of genes are on the same chromosome, what happens during meiosis. So if this goes through meiosis, basically you're going to produce gametes of 2 types. Both of the dominant alleles are going to be formed in this gamete, and the recessive alleles are going to be present in this gamete. But let's do the same thing we did last time. We'll flip these chromosomes around and go through meiosis again. So when we flip these around, and if we go through meiosis again, basically you just produce exactly the same kinds of gametes. All you've done is you flip both alleles around, so both dominant alleles are again facing the same pole. So again, half will be of this type where you have both of the dominant alleles of the 2 traits, and in the other half will be of this sort where you have both of the recessive alleles of the 2 traits we're looking at. So in this case now, it's a little bit different of an outcome because these are the same, as I mentioned, and these 2 are the same, and because you have these are the same and these are the same, you end up producing gametes of 2 different types and in equal proportions in the way I've drawn this out. Okay, so this phenomenon where a pair of genes do not seem to assort independently, but instead alleles tend to segregate together is called linkage. And another thing I want to sort of bring up is I actually say genes on the same chromosome do not necessarily assort independently. And I wasn't random about choosing this word necessarily. As it turns out, they do sometimes assort independently, even when they're on the same chromosome. But we'll come to that a little bit later in the lecture. Okay. So in a test cross of dihybrid, based on what you've learned up till now, you expected it to be a 1:1:1 ratio. But as it turns out, it's a 9:1:9 ratio. But based on the diagram I just showed you, it should be a 10:0:0:10 ratio, right? What's the deal with these two ones? Sometimes they do produce plants where the two pairs of alleles appear to be segregating somewhat, at least independently from one another. So why are we seeing all four types? In my diagram, I only show you two types, right? And that's what we're going to spend some time talking about. And the other thing that we will talk about, and I'll get to this towards the end of this lecture, why do we see this in this particular ratio? As it turns out, this 9:1:9 ratio is not a feature of linkage. It's a specific feature of the pair of genes we're studying here. So genes that are linked are not always going to show a 9:1:9 ratio, but it's going to be something different than the 1:1:1 ratio. Okay, so why in this ratio? And again, this is just to sort of illustrate the point I'm trying to make, which is the 9:1:9 ratio isn't universal. It just happens to be a specific feature of the pair of genes we're looking at. I'll come back to this later as well, not in today's lecture, but in another lecture to explain this. Okay, so this is just the observed ratio for this gene pair. So as I told you in the very first slide, it was Alfred Sturtevant that first correctly interpreted linkage. And as I also mentioned, Alfred Sturtevant was an undergraduate working in Thomas Morgan's laboratory. And the model system they used in all this work was the fruit fly Drosophila. So we'll begin our discussion of linkage using fly as an example. So before I get into the example, I need to say a little something about nomenclature for Drosophila genes. So the traits we'll be looking at in our example are the eye color trait and the wing shape trait. So wild-type flies have red eyes, and they have the normal shaped wings that you expect to see from a fly. And the two mutant traits we're going to look at, the mutant phenotypes, are purple-colored eyes and these really kind of shrunken or vestigial wings. And in the past, when I've given this lecture in front of a large group of students, a lot of times the students in the back would tell me that they had a hard time distinguishing the purple-colored eyes from the red-colored eyes. So I've added a little graphic to make sure it's very clear when I refer to the purple-colored eyes which flies I'm talking about. So there's the graphic that I've added to my slides. Okay, in Drosophila, genes are named according to their mutant phenotypes. And in the case of the mutant phenotypes we're looking at here, the purple-colored eyes and the vestigial wing shape, the names of these genes is the purple gene and the vestigial wing. And genes are in lowercase if the mutant phenotype is recessive to wild type. We put a plus next to the gene or the allele that corresponds to the normal wild type allele. So in our example, the purple gene, the purple mutant that confers purple-colored eyes is recessive to the wild type allele for eye color trait. So the gene is in lowercase, and the wild type allele is given a plus sign. And likewise, the vestigial trait is recessive to the wild type wing shape trait. So the mutant gene is designated in lowercase, and the wild type allele is given a plus sign next to the name of the gene. So the genes are in lowercase, so the mutant phenotype is recessive to wild type. Now, if I ask you to tell me what the genotype of these flies were, for the ones shown at the bottom, the ones with purple eyes and vestigial wings, based on what I've told you, you should be able to tell me exactly what the genotype is. So because these exhibit the mutant phenotypes, and the mutant phenotypes are recessive, we know that they must be homozygous for both the recessive alleles of these genes. And now the upper flies, the ones that have red eyes and normal shaped wings, it's less clear what their genotype is. They could be heterozygous for both of the dominant alleles, or they could be homozygous. So we really don't know. So we designate their genotype as follows. Okay, so let's talk about a cross that Sturtevant carried out using the purple and vestigial genes in one of his experiments. So basically what Sturtevant did is he took a wild-type fly, and it was a true breeding wild-type fly. And whenever I say true breeding, what that means is it's homozygous for all the alleles that are present in that organism. So it's homozygous for the wild-type alleles of the purple gene, homozygous for the recessive alleles of the vestigial gene. And he did a test cross, and a test cross parent is homozygous for the recessive alleles of the genes that were under study in this experiment. So it's homozygous for the recessive alleles of purple and the recessive alleles of vestigial. So he did a cross, and the only kind of gametes that the wild-type fly can make, based on what I've told you, is gametes that have both the dominant alleles of purple and the dominant allele of vestigial. And then the only gametes that these homozygous recessive fly can make are gametes that have the recessive alleles of the purple and vestigial genes. Okay, then you generate the F1 dihybrid, and its dihybrid, the genotype of the dihybrid is as shown, where it's heterozygous for both the purple and vestigial genes. So it receives the purple plus and vestigial plus from the wild-type parent, and it receives the recessive alleles of these genes from the homozygous recessive parent. Okay, so basically he did sort of, and then took this dihybrid fly, this F1 dihybrid, and he performed a test cross with it. And if I was giving this this lecture to you in person, I would ask you to tell me what the test cross genotype is, because I'll be talking about test crosses for a number of times in this and the next lecture. And again, the test cross parent it's homozygous for the recessive alleles of the genes that are under study. So this is the test cross parental genotype. So he performed this cross between the F1 dihybrid and a test cross parent. And the only kind of gametes that the test cross parent can make are gametes that have the recessive alleles of the purple and vestigial gene. So those are the gametes that would be contributed by the test cross parent. Whereas the dihybrid, the F1 dihybrid, could produce 4 different possible kinds of gametes, right? So it could produce these 4 possible gametes. So it's all possible arrangements of these alleles. And then I'm going to leave it to you. I'm going through some of this pretty quickly to get to the points I'm really trying to make, because a lot of this should just be a review of things you guys have talked about in the past. But basically, the phenotypes of these flies would be as shown below. It's just, again, it's combining, this is just a Punnett square essentially, and you should be able to infer what the phenotype should be based on things I told you in the previous slide. And if you expected independent assortment to occur here, what you'd expect to see is these offspring categories occurring in a 1-to-1 to 1-to-1 ratio. That's what you'd expect. If the purple and vestigial genes were on separate chromosomes and independent assortment was occurring, they'd all be seen in a 1-to-1 to 1-to-1 ratio. But what was actually observed by Sturtevant was the following results, where you can see that the offspring numbers varied tremendously. 2 of them, the number of offspring they found was somewhere in the range of 1,300. And then for 2 of the offspring categories, they were more around 150, basically. So this is not a 1-to-1 to 1-to-1 ratio. Although you could apply a chi-square test to really sort of do that, to really confirm that this is not independent assortment. And we'll talk about how you would use the chi-square test to do that a little bit later in this lecture. But for now, you can just eyeball this and see that this is not a 1-to-1 to 1-to-1 ratio. Okay, so what's going on here? So what we're going to do on the next slide is we're going to take a closer look at what's going on with the chromosomes in the dihybrid parent, this particular parent that I've circled here. So we'll look closer at the chromosomes to kind of say what's going on. So as the title of this slide indicates, this is hopefully a review of how recombination occurs for you. And the reason I say it's hopefully a review is because I know that this is covered in some of the introductory biology courses here. But I'll make no assumptions. I'll go through it fairly quickly because it should be a review, but I'm just going to go through it all just to make sure we're all on the same page. Okay, so this is the arrangement of alleles in the dihybrid parent that we were considering on the previous slide, where both the dominant alleles of purple and vestigial are on one homolog, and the recessive alleles are on the other homolog. And just to define a few terms also, I should probably make this clear. So basically, the blue and the pink colored chromosomes are homologs. Okay, these are chromosomes that are almost identical to one another. And then also these two thin blue strands are called sister chromatids. So, and each chromatid is a double-stranded DNA molecule. And likewise, the two pink strands are actually also sister chromatids, each of which is a double-stranded DNA molecule. Okay, so the first thing that happens during recombination is that there's what's known as nicking at corresponding locations on both homologs. So one of the sister chromatids, one of the blue ones and one of the pink ones is nicked. And by nick, I mean the DNA is actually cut at those sites. And the cut occurs at exactly the same location on the blue sister chromatid as the pink sister chromatid. And after the nicking occurs, there's strand exchange where the blue strand gets covalently attached to the pink strand and the pink strand gets covalently attached to the blue strand. And the strands are resealed basically, so there's no longer a cut. Okay, at the next stage what happens is the spindle attaches to the centromere and it pulls the two homologs apart from one another. So the homologs pull apart, cell division occurs, and these are the products that you would see. So now what you have is, if you look at there's two different cells here. If you look at the upper cell, you can see that the purple plus and vestigial plus alleles are present on one of the sister chromatids, the blue sister chromatid, whereas the recessive purple allele associated with the pink chromosome is on the lower sister chromatid, whereas the dominant vestigial plus is still associated with the blue chromosome. Okay? And likewise, you see the reciprocal products in the other cell product, the other product of cell division. All right, so then we go through the next step of meiosis, where these cells divide to produce the gametes. And basically what you end up with is the four following gametes. And we're going to now sort of name the types of gametes these are. So two of these gametes have the parental arrangement of alleles, and we'll call these the parental gametes. And by parental arrangement alleles is if I put up again the genotype of the dihybrid parent that produced these gametes. So here's the genotype of the dihybrid parent. What you can see is the gametes that I'm calling parental, the arrangement of alleles are exactly the same in the parental as in the dihybrid parent. So you have one chromosome that has both the dominant alleles of purple and vestigial, and that's one of the parental types. And the other parental type is a gamete that has the recessive alleles of purple and vestigial. So again, these alleles are the same as in the parental. And the other two products of meiosis we call non-parental or recombinant types. And the reason we're calling these non-parental is because the arrangement of alleles on these chromosomes is different than in the parental, right? So you have a recessive allele, the purple, and a dominant allele, the vestigial, in one of the non-parentals. And you don't see that arrangement of alleles in the parental configuration. And the other non-parental has the dominant allele of the purple gene and the recessive allele of the vestigial gene in the gamete. So again, these do not look like the chromosomes that are present in the parent that gave rise to these gametes. So one recombination event gives rise to two recombinant products and two parental products, or non-recombinant. And the reason I'm illustrating that point here is because I'm going to talk about other types of crossover events that occur between the loci that we're thinking about, because it can give rise to other kinds of recombination products. Okay, let's consider some other types of crossovers, and we'll consider how these crossover events result in the kinds of gamete types that they result in. All right, the first one we'll consider is the following. And please note that when I go through these, I'm talking about the number of crossovers that occur between loci of interest, okay? So here's the first one. When I give this lecture in person, I usually ask people how many crossovers occur between the loci of interest, and oftentimes people will say 1, but in fact the correct answer is 0. Because again, we're only concerned about crossovers that occur between the loci of interest, so those that occur between the purple and vestigial genes in this case. And then what are the genotypes of the resulting gametes? The genotypes look like this. And then if I ask you to look at the the gamete genotypes and tell me whether these are parental, non-parental. I think if you look closely, you can see the arrangement of alleles on all the 4 gametes that are shown here look like the parental types. So this results in 4 parental gametes. All right, let's consider another type of crossover. This is how many crossover events are occurring between loci of interest. In this case, there's 2. And the 2 crossovers only affect 2 chromatids. So if you look at this, the outer blue sister chromatid and the outer pink sister chromatid, they're not affected by these crossover events. It's just the 2 inner ones. So this is a double crossover affecting 2 chromatids. Now let's look at the effects of these crossovers on the resulting gametes. These are the resulting gametes that you'd expect to see. And if you look at the arrangement of alleles in the gametes, and I ask you, what's the result of this in terms of whether they're parental or non-parental, what you can see is, once again, we're producing exactly the parental arrangement of alleles in all of the gametes. So again, all 4 of them are parental. So to really make sure that you understand what I'm saying, what you have to do is you have to think through what happens when you pull these chromosomes apart, pulling at the centromere, and you just follow the chromosomes and see what alleles are going with which of the chromosome products. So it's hard for me to illustrate that here on these slides, but you have to that's the way you're going to have to try and make sure that you understand what's going on here. Okay, so now we'll consider another set of crossovers. And in this case, we're again considering 2 crossovers, but this is 2 crossovers that now affect 3 chromatids. You see only one of the chromatids here, in this case, the upper blue sister chromatid, is not affected by these crossovers. So again, this is 2 crossovers between the loci of interest that affect 3 chromatids. So now if we look at the products of recombination in this case, what you can see is, in fact, there's something different going on in this case. So if you look closely at the parental or the arrangement of alleles, what you can see is there's 2 gametes that are produced that have the parental arrangement of alleles, but 2 of the gametes have the non-parental arrangement. So the ones that have the non-parental arrangement are the ones with the recessive purple allele and the dominant vestigial allele, and the one with the dominant purple allele and the recessive vestigial allele. So this kind of double crossover involving 3 chromatids results in 2 parental and 2 non-parental gametes. And the last one we'll consider, and we could keep going. I could do 3 crossovers, I could do 4 crossovers, but I'm going to stop with, I'm just going to do 2 double crossovers here. So here's another double crossover, and in this case, it's all 4 chromatids that are actually affected with the double crossover. So 2 crossovers, 4 chromatids affected, and now if we look at the genotypes of the gametes that are produced, it turns out all 4 of the gametes that are produced from this type of crossover event, double crossover I should say, are all non-parental. All of the gametes have the non-parental arrangement of alleles. So you result in 4 non-parentals. Okay, so I want to make sure that you understand all of this. So this is something you have to go through in your own mind and kind of help pull the chromosomes apart to make sure you can see how the different alleles are distributing. Before we get too far along on our discussion of linkage, I have to introduce you to some notation and vocabulary. So what I'm showing you here on this slide is I'm showing you the arrangement of alleles in the dihybrid parent we've been considering on the last couple of slides, where I've got the recessive alleles of the purple and vestigial gene on one homolog and the dominant alleles of purple plus and vestigial on the other homolog. So one way I could designate that is as shown here, where the 2 straight lines represent the 2 different homologs, and the straight lines themselves indicate that these 2 genes are linked. The purple and vestigial genes are linked to one another. But I could also use a more lazy shorthand notation to designate exactly the same thing by drawing this out as follows, where I'm not drawing out both the homologs, but just the single line indicates that the recessive alleles of purple and vestigial on one homolog and the dominant alleles of purple and vestigial are on the other homolog. So these things mean the same thing, and these lines just indicate linkage, that these genes, purple and vestigial, are linked to one another. I could also use a slash instead of a horizontal line. So for example, the genotypes I'm showing you at the top of the slide could be represented as shown at the very bottom, where I have the purple vestigial alleles on one homolog And then the slash designates that I'm now considering the other homolog. The purple plus and vestigial plus are on the other homolog. So this is another even more shorthand notation for designating the genotypes that are shown above. And the other issue I need to introduce you to is the phase of the alleles. So if the dominant alleles are on the same homolog, they're said to be in the cis configuration. And the phase just refers to the allele orientation or the allele arrangement. So again, if the dominant alleles are on the same homolog and the recessive alleles are on the other homolog, this is said to be the cis configuration. The trans configuration would be one where, for the two genes we're considering, if one homolog has one of the dominant alleles and the other homolog has the other dominant allele, that's said to be the trans configuration. So an example of that is shown on the very far right. Okay, and this actually knowing the phase is very, very important in being able to predict the outcomes of a cross, as you'll see later on in this lecture. One last notation that I need to sort of introduce you to is the semicolon. And the semicolon indicates if a set of genes are on separate chromosomes. So the white gene and purple gene are on completely different chromosomes. The semicolon indicates that these are different chromosomes altogether. All right, I have a question for you. A man is a carrier for sickle cell anemia and Tay-Sachs disease. Let's say the genes involved in both these recessive disorders are on the same chromosome. His wife has sickle cell anemia, so that tells you she's homozygous for the recessive allele, and she's also a carrier for Tay-Sachs, and they have a child with both diseases. Is their child a product of a recombination event that took place in the father? So we're only really considering what's happening in the father. I'm going to give you three choices to choose between: yes, no, or I don't know. So as I said, I'll just let you pause the video here and you can think about it. And when you're ready to answer or see the answer to the question, just hit start again. But I'm only going to pause for a few seconds. All right, for the first thing we have to think about is, what is it that we need to know about the father to really answer this question? And I'll just tell you right up front, the answer is we don't know. So what do we need to know to answer this question about the father? So we know that both the mother and the father donated recessive alleles to their offspring for both of these traits. But the father's heterozygous for both of the traits. So there's two different possible arrangement of alleles of these genes in the father. So the one that I'm showing you here on the left shows the cis configuration, and the one on the right shows the trans configuration. So again, the father has transmitted both recessive alleles to their offspring. So if the father is in the cis configuration as shown on the left, then the father did not, no recombination had to take place to transmit these alleles to the offspring. However, if the father is in the trans configuration as shown on the right, a recombination event had to take place for the father to transmit both recessive alleles to their offspring. So hopefully that's clear to you. Now we're going to talk a little bit about identifying parental type in a double heterozygote. This is actually very important in interpreting the outcome of a cross, or in predicting the outcome of a cross if we know something about the arrangement of alleles in the parent. So one option for identifying parental type is knowing what gametes produce the heterozygous F1 parent. So let's imagine in this example, we know that one of the parents is homozygous for both the dominant alleles of the purple and vestigial gene, and the other parent that we're using to produce the F1 parent is homozygous for the recessive alleles of the purple and vestigial gene. So in this particular case, there's only one kind of gamete that each of these parents can make. So when these parents mate and produce offspring, there's only one kind of offspring that you can make. And that's the following. The F1 offspring has to be an offspring that has both the dominant alleles of these two traits on one homolog and the recessive alleles on the other homolog. And this is also called the cis configuration, right? Because the dominant alleles are on the same homolog. This is the only possible outcome from this kind of cross, because the dominant alleles are coming from one parent and the recessive alleles are coming from the other parent. All right, we're going to consider now the same alleles, but the parent genotypes are a little bit different. So in one case, one of the parents is homozygous for the dominant alleles of purple and the recessive alleles of vestigial, and the other parent is homozygous for the recessive allele purple and the dominant allele of vestigial. Once again, there's only one kind of gamete each of these parents can make. The parent on the left can only make gametes that have the dominant allele purple and the recessive allele vestigial, and the parent on the right can only make gametes that have the recessive allele purple and the dominant allele vestigial. And so when these parents produce offspring, the offspring genotype is going to look as follows, where one of the homologs has the dominant allele purple and the recessive allele vestigial, and the other homolog has the recessive allele purple and the dominant allele vestigial. So now the dominant alleles are on different chromosomes, and so this is the trans configuration. So a really interesting thing about this is if I asked you what the genotype of these two flies in these two examples that we generate, these two F1 offspring were, the genotypes would be exactly the same. In both cases, their genotypes are heterozygous for both the purple and vestigial genes. So the genotypes are identical in these two parents. However, the phase of alleles in these two parents is different, okay? You can't see that phase by just drawing out the genotype. You have to do something else to know what the phase is. So the other option for knowing the parental type in a double heterozygote is by doing a test cross and looking at the relative frequency of the progeny phenotypes. And probably the best way to illustrate that is by just going back to one of the crosses we talked about earlier that Sturtevant first did when he first established linkage. We again use the purple and vestigial genes. So one thing that I didn't really emphasize, but if we go back to a previous example that I'm going to emphasize now, is when you take this dihybrid parent and you do a test cross, what you see is that you see a lot of two different categories of offspring, and you see much fewer of two other categories. And in fact, the ones that predominate are in these categories turned out to be the parental type. So this is the parental type, red eyes and normal wings, and purple eyes and vestigial wings. These are the parental types of it was parental type gametes that are producing these offspring, and they're roughly in equal proportions. And then the other category are the recombinant types. They're in lower numbers, and they're also the two recombinant types are roughly in equal numbers to one another. So these are the gametes that give rise to these different, in the F1 parent, that give rise to these offspring. So the parental gametes are more common than recombinant if the genes are linked, okay? So, and the reason that's the case is because when you produce gametes, most of the gametes that you make are not going to have undergone recombination between the traits you're looking at. So most of the time when you make gametes, the gametes are going to have the parental arrangement of the alleles because it's only the occasional time of recombination that takes place that you produce recombinant gametes. So again, mostly you're going to see the parental types being produced, and again, they're the same as the gametes that made the parent. And occasionally you're going to see a crossover event that takes place between these alleles of interest. And so when you do see that, you'll produce a couple of recombinant types, okay? Or non-parental. Now we'll work through an example together to reveal how you can identify a parental type in a double heterozygote by doing a test cross and looking at what the two most abundant progeny types are. And again, this only works if the genes are linked to one another. If they're not linked, the results will just look like independent assortment. So let's say we do the following test cross where we have a dihybrid parent, heterozygous for both the purple and vestigial genes. We don't know the phase of alleles in the test cross parent, and we're going to do a test cross. So we're going to cross it to a homozygous recessive. And then we'll consider, so we do this kind of a cross, we're going to see 4 different types of offspring. And let's say the 4 offspring that we see are in the given numbers that I've shown below, where flies with red eyes and vestigial wings, we see 1,287 of those. I won't go through all of them. You can see the numbers next to it. So, okay. So if we consider what kind of gametes were transmitted to each of these progeny flies by the test cross parent, the parent that's homozygous for both recessive alleles, there's only one kind of gamete they can transmit, and that's basically the recessive alleles of the purple and vestigial gene. So one of the homologs has the recessive alleles of the purple and vestigial gene for all these flies. And then it's just a matter of looking at the gene or the phenotypes of each of these offspring to infer what other combination of alleles was inherited from the dihybrid parent. So, for example, the flies with red eyes and vestigial wings, that fly must have inherited the dominant allele of the purple gene because it has red eyes, and the recessive allele, the vestigial gene, from the test cross parent. And the flies to the right of that one, the ones with purple eyes and normal wings, that must have inherited the recessive allele, the purple gene, and the dominant allele, the vestigial gene, from the dihybrid parent. So, in other words, these are the homologs that must have been inherited. So you can also look carefully at this and see that these two flies, the ones at the top, are roughly in equal proportion. They're both roughly 1,200 to 1,300 flies. And they're by far the most abundant category. The ones below them are also roughly equal numbers, 170 and 154, but they're much smaller numbers. So this tells us right up front that the arrangement of alleles in the dihybrid parent must have been in the trans arrangement, because one of the parental types is purple plus vestigial, and the other parental type is purple vestigial plus. And then I think I'm not going to belabor the point, but you should be able to work out what the recombinant types are just from the phenotypes of the flies shown below. But basically, one of the recombinant types is going to be the dominant allele of purple and the dominant allele of vestigial, and the other recombinant type, the one at the bottom right, is going to be the recessive allele of purple and the recessive allele of vestigial. So those are the recombinant types. So this is a really good problem because it helps to illustrate some of the principles I'm trying to get across. So I'll read this through to get you set up. The pedigree shows segregation of two linked disorders. One is autosomal dominant, where big A equals the disease and little a equals not diseased. And one is autosomal recessive, where little b is the disease allele and big B is the not disease allele. And the question we're asking here is the gamete that 3-1 received from 2-2, so is the gamete that this individual received from his father, is it parental or is it non-parental? Okay, that's the question we're asking. And to sort of help you along, I've defined the different, what the phenotypes mean with respect to the two genes we're talking about. All right, so the first thing you need to work this out. Now, normally if I was in a lecture, I would say, go ahead and break down the question, talk to your neighbors, and come up with a systematic step-by-step strategy to solve the problem. Now, of course, if you're listening to the lecture with a friend that's taking the class, it's totally fine with me if you want to talk to your friend that you're listening to the lecture with to work out how you're going to figure out how to answer this question. But I'm also going to give you a series of steps you can follow to answer the question. You're perfectly welcome to pause the lecture now and see if you can do it all on your own. You can look and see what my suggested strategy is for solving this problem, and then work it out. It's up to you. So you can pause it now if you'd like. But I'm going to keep going and give you my tips for how to solve this problem. And then at that point, if you want to pause the lecture and answer the question, that's fine as well. So the way I would go about this is, in the first step, I try and figure out all the genotypes, kind of looking at what the phenotypes of these individuals are. I try and figure out what their genotypes must be. The next step would be to figure out what gametes that made 2-2. So what are the gametes that made this individual 2-2? Because that will help you determine what the phase of the alleles is in this individual. And then the next step would be to figure out what gamete from 2-2, what gamete that individual gave to produce his son, 3-1. And then finally, based on the gamete that this individual transmitted to his offspring and then what we know about the phase of alleles in this individual, we can ask whether the gamete that 2-2 gave to his son have a different genotype than the gametes that he received. Okay? So you can pause the lecture here and try and work this out on your own. But I'm just going to continue and show you how I went about doing this. And and you can just use that to check your work to see if you got the right answer. Okay? Hopefully you've had a chance to try and work this out on your own. But in any case, if you just want to check your answer, or if you'd like to go through the logic, we can just now work through this together. At any point, you can stop the video if I've given you a few hints to get started, and just try and complete the problem on your own. So what we're going to do here is we're going to try and start off by figuring out what the genotypes of all the individuals in the pedigree are. And we'll start with individual 1-2 because that's one of the easiest ones to identify. And we're going to finish by trying to figure out what the genotype of 2-2 is, because not only do we know the genotype, we need to know the phase of the alleles. All right, starting with 1-2. So we see this person actually has the autosomal recessive disorder, but does not have the autosomal dominant disorder. So this person must be homozygous for the little a allele and homozygous for the little b allele. So this person must be homozygous for the A and the B genes. And the next one we'll try and figure out is individual 1-1. So this person has the autosomal dominant disorder, but does not have the autosomal recessive disorder. So what we know is this individual must have dominant alleles of A and dominant alleles of B, but we don't know if this person's homozygous for these alleles or if they're heterozygous. So this is the genotype of this individual. And now we'll do individual 2-1. So 2-1 does not have either of these traits. So that tells us right off the bat that this person must be homozygous for the recessive allele of the autosomal dominant disorder. So there must be an AA homozygote. But this person also does not have the recessive trait, or the recessive disease. So this person must carry at least one dominant allele of the B gene. And if we also look at individual 3-1, this is the offspring of individual 2-1 and 2-2, we can see that this individual has the autosomal recessive disorder. So they must have gotten an allele of the B gene from individual 2-1 and 2-2. So this person must be heterozygous for the B gene. So they have one dominant allele because they don't have that trait, and they have a recessive allele because they pass that allele on to their offspring. All right, so the next genotype we'll try and figure out is individual 3-1. So 3-1 has both of the traits, the dominant and recessive. So right off the bat, we know they're homozygous for the recessive trait allele. So they're homozygous for BB. And we also know they have at least one of the dominant alleles for the dominant disorder. So they have one of those, which they must have obtained from individual 2-2, who has the dominant disease trait. And they must be heterozygous for the A gene, because individual 2-1 does not have, it only has recessive alleles of the dominant disorder to transmit. So the genotype of individual 3-1 is as follows. They're a big A, little a heterozygote, and a little b homozygote. Now we're going to try and figure out what gamete individual 1-2 gave to individual 2-2. And that's pretty simple to figure out because individual 1-2 only can transmit one type of gamete. The only kind of gametes individual 1-2 can make is a little a, little b gamete. So they must have given individual 2-2 a, little b gamete. And now we'll figure out what gametes individual 1-1 transmitted to individual 2-2. So because 2-2 has the dominant trait, they must have inherited the dominant allele of A from individual 1-1. But this individual also does not have the recessive trait, so they must have inherited the dominant allele of gene B from individual 1-1. So this is the gamete that individual 1-1 must have transmitted to individual 2-2. So the gametes that 2-2 received are big A, big B, and little a, little b. And we now know the genotype of individual 2-2. This individual is an AA heterozygote and a big B, little b heterozygote. However, we know more than just the genotype. We also know the phase of the alleles because we know what gametes produced individual 2-2. So we know that the actual phase of the alleles is as shown, where one of the homologs has the big A and big B alleles, and the other homolog has the little a, little b allele. So this is in the cis configuration. All right, so now we're in a position to really kind of address the ultimate question here, which is what alleles individual 3-1 inherited. So first we'll figure out what alleles 2-1 gave to 3-1. So this individual must have given a little a allele, because that's the only kind of allele that they have at the A gene to transmit. And because this person does not have, or has the recessive disorder, they must have also received a little b allele from 2-1. So the gamete that 2-1 gave to 3-1 is a little a, little b allele, or little a, little b gamete. And the gamete that 2-2 gave to 3-1 must be a big A, little b allele, because this individual has the dominant disorder and the recessive disorder. So the gamete that the gamete that 2-2 gave to 3-1 must be a big A, little b allele. Okay? And that's the phase of the alleles in individual 3-1. So the gamete that 2-2 gave to 3-1 is a big A, little b gamete. And so ultimately the question we were asking here is the gamete that 3-1 received from 2-2 a parental or non-parental? And if you look at the parental arrangement of alleles, which are in the trans configuration in 2-2, you can see that the gamete that was transmitted is a big A, little b allele. So this must be a recombinant or non-parental gamete that was transmitted. So the answer is a non-parental. All right, just to summarize some of the things I've been telling you up till now. Basically, genes on the same chromosome can show linkage instead of independent assortment. And the gametes that are produced mostly have the same allele combination as the homologs in the parent. Okay, that's the parental types. Recombination can give rise to gametes with both non-parental or recombinant types, allele combinations. And the two parental types are more abundant and roughly equal to one another, and the two recombinant types or non-parental types are less abundant and roughly equal to one another. Okay? All right, so as I mentioned at the beginning of this lecture, Alfred Sturtevant is actually the person who had the major insight that led to the idea that these odd results from these crosses he had been doing might be explained by the fact that these genes that he's looking at were actually on the same chromosome, not on separate chromosomes assorting independently. And as the story goes, Sturtevant stayed up most of one night working out the details of linkage analysis instead of doing his homework for the evening. So I think that story is true. But here's the insight that came from his work. Basically, the major insight was that if crossovers occur at random along the length of the chromosome, then the probability of a crossover event occurring between any two loci is proportional to the distance between them. So if the distance between them is really large, then the odds of a crossover event occurring between them would be high. And if the distance between two genes is very small, then the odds of a crossover event occurring between them is very low. So just to illustrate this point, we'll consider a couple of genes. So for example, let's consider genes A and B. Let's say that they're very widely spaced from one another on the chromosome. So if they're widely spaced, then the odds of a crossover event occurring between them are fairly high. So in this particular example, there's two crossover events that occur between A and B. Now we're going to drop down a third gene, D, and we'll put D really close to B. So if we consider the same three set of crossover events, you can see that there are no crossover events that occur between B and D, and the odds of that happening are much lower because B and D are very closely spaced to one another. So in other words, the crossovers between A and B are much more likely because they're distantly spaced genes, and crossover events between B and D are much less likely because those genes are very close to one another. And Sturtevant used this basic ideology to develop what's known as a genetic map, and we'll talk about that a little bit on the next slide. So, and actually Sturtevant went a little bit further to calculate the frequency with which crossovers take place between the gene pairs that he was studying. And again, I'll go back to the example from last time. the cross is something like this. Dihybrid for the purple and the vestigial genes, you're doing a test cross. And let's say you get these progeny class categories. So again, we know just right immediately that there's that this involves a pair of genes that are linked to one another, because you're not seeing them in 1-to-1 to 1-to-1 proportions, and you see that there's two progeny categories that are roughly equal in their number to one another and way more abundant than the other progeny category. So this is the parental type then, and this is the recombinant type. And so the way he determined, he calculated how far apart these genes was by calculating the fraction, or the frequency at which recombination takes place between these genes. And basically the way he did this, he sort of said, well, from this cross, there's 1,287 plus 1,204 parental types, and there's 170 plus 154 non-parental types. Okay, so these are the parental types, again, the most abundant, and the other type is the non-parental type, the least abundant. And the parental types are purple plus and vestigial, and purple and vestigial plus, and the non-parental types are the other category, purple plus, vestigial plus, and purple, the recessive alleles of both of these traits. And then so what sort of decided was maybe the distance these genes are apart, the purple and the vestigial genes, is related somehow to the percent recombination that occurs between them. So he decided, I'll just calculate the percent recombination that occurs. So what he did is he totaled up the total number of recombinant types and divided that by the total number of progeny that he observed, and he multiplied by 100 and calculated that was 11.5%. So, and he gave rise to a term that was called map distance. So he said these genes map 11.5 map units apart, and then the term map unit was also given the name centimorgan. So 11.5 map units is also equal to 11.5 centimorgans in honor of the lab in which he did all this work. So 11.5% recombination equals 11.5 map units equals 11.5 centimorgans. They all mean the same thing. Okay, so I have another problem here. In corn, yellow kernel color is dominant over white. Normal endosperm is dominant over shrunken endosperm. You have a dihybrid plant that is test cross, and you produce the following progeny. So you know what the test cross parent genotype is. It's homozygous recessive for both the kernel color trait and for the endosperm trait. And you receive the following results. So I've got two questions for this. The first question is, what are the parental types of the dihybrid plants? And the second question is, what map distance separates the Y and SH genes? So you can stop this lecture right now, and you can work on the problem if you want, or you can just, well, actually that's what I encourage you to do. And then I'll just go through and tell you what the answers are. So the parental types are these two here, because again, what I told you is the parental types are the two most abundant types. They're roughly in equal proportions, so these are the two most abundant types. One type is yellow and shrunken. Yellow is the dominant Y allele, shrunken is the recessive SH. So one of the parental types is the dominant Y and the recessive shrunken, and the other parental type is white, which is the recessive allele for Y, and normal endosperm, which is the dominant allele for the endosperm trait, Y and SH. So those are the parental types. And then the next question is, what map distance separates the Y and SH genes? So one of the things that gives you a clue right from the start that rules out one of the answers is, one of the answers I said is the Y and SH genes are unlinked. So the fact that you see two progeny categories that are abundant and similar in number, and two other progeny categories that are lower in number and roughly equal in proportions, that's an indication right from the start that linkage is involved. So that's probably not the right answer. And then in terms of the map distance, the way we calculate that is we take the total number of recombinants, which are the two less abundant categories, 106 plus 114, and divide that by the total number of progeny that we see. And if you do that, you should end up with a map distance of 22 centimorgans. All right, so the last thing we're going to talk about here is how we identify whether linkage is occurring versus independent assortment. And the statistic test we do to determine this is one that should be familiar to you. It's the chi-squared test. And since we've been through all of this, I'm just going to go through this problem fairly simply. The critical things that we need to be considering here is, what's the null hypothesis in our experiment here? So one of the difficulties with determining the null hypothesis with linkage is that if the genes are linked, you don't actually really know what the map distance is that separates the genes. And to use a chi-square test, you have to have a very specific prediction of your outcome. So in this case, the null hypothesis is that these genes are assorting independently because we know what the predicted outcome for independent assortment is. And if the null hypothesis is rejected, then that tells us that it's probably linkage that's operative. Okay, so the null hypothesis is, again, more formal terms, the ratio of offspring is consistent with what we would expect if the two genes were assorting independently. So that's the null hypothesis. So again, let's say we did a cross and we found the following results. And again, we're using the familiar traits, the eye color trait in flies and the vestigial wing trait. And let's say we do a cross between these traits and we got the observed numbers of progeny, right? So what you can do is you can look at this and say, well, we see more of two of the categories than we do of two of the other categories, and the two that we see that are more of are roughly equal in number, and the two that we see less of are roughly equal in number. So this is like a hint that maybe linkage is involved, but the numbers are not so different here than some of my previous examples. So you wouldn't look at these and necessarily say right away that linkage is involved in this, what's going on here. So we'll do the chi-squared test to sort of test whether linkage is involved. And basically, again, the null hypothesis, these genes are independently assorting. There's 1,000 total progeny that we counted. And if it's independent assortment, we'd see an equal number of each of them. So that means 250 of each of them. And then the rest of these are just arithmetic operations, right? The observed minus the expected squared. So that's these values here. Then the observed minus the expected squared over the expected. So for this first one, it's 2.5. And we're just going to use the same kind of math to determine what that is for the rest of these. I assume you guys can do all this arithmetic. And then we have to do is we have to tally up this number to get a total. And the total is 12.2. And then from this, we actually have to consult the chi-squared table, which is on the next slide. And we basically look at the closest number in this chi-squared table. But we have to determine the number of degrees of freedom. And really, again, the degrees of freedom is the number of independent variables minus one. It basically, the more independent variables you have, the more chance, well, that's a bad choice of words, the more opportunity for chance to occur and give you a fluctuation from the observed outcome. So in this case, there's 4 different progeny categories. The number of degrees of freedom is the total number 4 minus 1, so it's 3 degrees of freedom. And then we find the closest value from our chi-squared calculations, and the closest value is here, 12.8. And basically, the corresponding p-value for that is 0.005. And I think what you guys were told in the first half of the class is that we reject the null hypothesis if the p-value is greater than 0.0 or smaller than 0.05. And so this is much, much smaller than 0.05. And again, this p-value refers to the chance you'd expect to see results of the observed type that we found in our experiments from the null hypothesis just by chance alone. And so this is a very, very low probability that we'd see a result that's that much skewed from the expectation from independent assortment from chance alone. So because that's a very, very low probability, greatly below 0.05, it basically just tells us that that we would reject the null hypothesis. So again, this is the probability of observing this data that the two genes are actually independently assorting is 0.0005. So that's a very, very low probability indeed. So we reject the null hypothesis. All right, the last thing I'm going to talk about in this lecture is something I'm going to leave you guys to do. I'll post the answer later, but I just wanted to set up the question and give you the sort of logic for solving this problem, and then I'll let you guys do this on your own. But again, I'll post the answer for you. So in a different plant species, elongated seeds is dominant over round, rough bark is dominant over smooth, and yellow flower is dominant over white. So you take a trihybrid, so you're heterozygous for all 3 traits. I haven't told you anything about the parental types, or this is just the genotype. You take this trihybrid and you test cross it with the following progeny, and these are the progeny you get. So I want you to take this information from these progeny and, as best you can, determine whether linkage is involved between any of these genes, and then also determine if linkage is involved, what the map distance is between the genes, and I want you to work out what the phase is in the cross okay? So I know this involves 3 traits. I haven't asked you to do that up to this point. I haven't described how to do this. So I'm just going to give you the logic and how to do this, and I'll let you work the rest of this out on your own. So basically, you just need to analyze these in a pairwise basis. So for example, you can start off by just looking at the seed shape trait versus the bark trait. So you look at those first of all. So you just basically look at just those traits and you group them together. So basically, what you can see is there's 9 plus 12 that have elongated and rough. There's 238 plus 237 elongated smooth. 243 plus 242 rounded rough, and 9 plus 10 rounded smooth. So I've just totaled those all up over here. So you just basically look at the gene pairs as just pairs individually to say whether these particular gene pairs, elongated rough traits that correspond to the elongated rough traits, show evidence of linkage, all right? And if they do, what the map distance is and what the parental types might be. And then once you've done it for those 2 traits, you go on to another 2 traits. So in this case, it's going to be the seed shape trait versus the flower color trait. And you just go through the same series of steps. You group things together, determine the totals, and use that to determine if linkage is involved. And then you'll do the same thing for the last 2 traits together, okay? So I'm going to leave it at that, and this is the end of the lecture. And good luck with the problem, and I'll post the answer at another time.